Dear students, in this module, I'm going to introduce you to protein structure prediction. You know that there are over 1 million proteins in the UniProt database, but there are only a few thousand in the PDB. So apparently, there is an orders difference between the number of sequences that we know for proteins and the number of structures for them. Why should such a situation be there? It's a very interesting question. The reason for such a situation is that determining the protein structure is an extremely complex problem. Moreover, most proteins do not allow crystallization. So therefore, if you cannot crystallize a protein, then you cannot obtain an X-ray crystallography for them. Of course, there are other advanced techniques such as the NMR, which you can then use to determine the structure of the protein. But once you have obtained the X-ray of a protein, even afterwards, to solve that structure is a very complex problem as well. So these are the reasons why we have so few structures in the PDB as compared to the sequences in UniProt. So given this huge difference in the number of sequences and the number of structures, is it possible that we can somehow predict using computational tools or bioinformatics to predict the structure of these proteins? Yes, indeed, that is possible. But to do that, we first have to see how the proteins structure themselves naturally, and then using that information, we can have a sequence of amino acids, and then we can fold it using that knowledge. You know that the basic idea is that amino acids, they determine the structure of the protein. So what if we are given the UniProt database and we see which amino acid is present in which specific secondary structure in the protein? So you can run a survey. The survey can include to register each type of amino acid in the different secondary structures within the protein. So once you have obtained such a list or such information, then you can actually predict which amino acid can go and form which secondary structure. Now, one important angle to this problem is, why do we need to predict? Isn't there an exact solution or a definitive answer to the protein folding? So given a sequence, can we somehow for sure predict which secondary structure will be formed? Well, sadly, the answer to that question is no. Till date, we have not been able to come up with a deterministic solution to the protein folding problem, and therefore all we are left with is to predict using the statistical information that is there in the known structures. So the proteins in nature, they fold either spontaneously or with the help of enzymes or chaperones. So for us to fold a sequence, we need to mimic this design. Towards mimicking this design, we need to know which amino acid has what kind of characteristics in the folding process. So the three important steps that occur given an amino acid chain towards the protein folding. Step number 1 is the collapse of the protein chain onto itself. So this collapse actually buries all the hydrophobic amino acids into the core of the protein. So once this collapse happens, the protein is somewhat folded and all the hydrophobic amino acids are there in its core. The second step is the formation of alpha helices and beta sheets, that is a fluid globule. And the third step is the compacting of this structure. So the proteins, once they have their secondary structures in place, they are compacted by the formation of hydrogen bonds. So once these hydrogen bonds or weak electrostatic interactions are settled, then the protein is compacted. So in conclusion, the protein structure prediction problem, it involves learning the behavior of amino acids during the folding process and then using this information to fold protein sequences which have an unknown structure. This information can be very useful in predicting the overall protein function as well.